What if you could figure out the entire blueprint of life just by turning off one tiny part at a time? It sounds like something straight out of science fiction, right? But it's actually a real, revolutionary tool in biology. And it all starts with some pretty unusual mice. All right, so take a look at these two. On the left, you've got your standard, everyday mouse. But the one on the right, well, it looks like it's been hitting the gym hard. I mean, seriously, it has more than double the muscle mass of its neighbor.
So what's going on here? How can you get such a dramatic difference between two animals that should be, for all intents and purposes, the same?
And here is the absolute kicker. Their DNA is practically identical. The only difference is that in our supramuscular mouse, a single gene has been completely switched off or knocked out. And that simple, powerful idea is what we're going to dive into today. This concept that you can finally understand what a gene does by seeing what happens when it's not there, it literally changed everything for genetic research. You know, for decades, scientists could read our genetic code. But that was kind of like having a huge, complicated instruction manual for a machine without knowing what any of the individual parts actually do. To really figure it out, they had to find a way to take out one piece at a time and just see what broke. And the tool they invented to do exactly that is the knockout mouse. So what exactly is it? Simply put, it's a mouse where researchers have gone in with almost surgical precision and silenced one specific gene. Then, by watching that mouse, how it looks, its health, its behavior, everything, they can deduce what that missing gene was supposed to be doing all along. Now, this was a massive leap forward. Before knockout mice, scientists had something called transgenic mice, where they'd add a new gene. But the problem was, it was messy. The new gene was just inserted randomly. The knockout technique was a total game changer because it was precise. It allowed for a loss-of-function study, letting them see what happens when a gene is taken away, not just when one is added. This technology was so incredibly transformative that it was recognized with the highest honor in science.
Yep, in 2007, the brilliant minds behind this breakthrough were awarded the Nobel Prize.
The prize went to three scientists, Mario Capecchi, Sir Martin Evans, and Oliver Smithies. It was really their interconnected work, from figuring out how to grow embryonic stem cells to the actual principles of gene targeting, that all came together to make the knockout mouse a reality. So how do they actually do it? Well, it's a pretty complex process, but here's the gist of it. They design a piece of DNA to target and disrupt a specific gene. They introduce that into mouse stem cells in a petri dish. Then, they inject those modified cells into a normal mouse embryo, creating what's called a chimera. And then, through careful breeding, they can eventually produce a line of mice where every single cell is missing that one gene. Okay, so we've got the concept and the recipe, but what's the real-world payoff? Why does this matter so much? Well, this is where it gets absolutely fascinating. Take a look at this. The mouse you see here is a knockout model being used to study human obesity. By comparing it to a normal mouse, scientists can get priceless insights into the genetic triggers and factors that contribute to this condition in us. And this is just a fantastic visual of how genes control traits. Scientists knocked out a single gene called FGF5, and boom, the mouse grew this incredibly long Angora-like fur. It's a direct, visible link showing that this gene's job is to regulate hair growth. And because the genetics of mice and humans are so incredibly similar, these knockout models have become absolutely essential for studying some of our most complex diseases. We're talking everything from cancer and heart disease to diabetes, Parkinson's, and even things like aging and anxiety. One of the biggest breakthroughs ever came in cancer research. For a long time, scientists suspected that a gene called P53 helped stop tumors from forming. So they created a mouse without it. And when they saw cancers developing, they had what they called the ultimate proof. This discovery totally changed how we fight cancer, revealing a guardian of our genome and paving the way for therapies that target this crucial defense system. But as powerful as this technology was, the original version had one major limitation. And solving that problem led to an even more advanced tool. So here was the problem. What if the gene you want to study is so essential that the mouse can't live without it? It turns out about 15% of these gene knockouts are lethal during development. The mouse never even gets a chance to grow up, so you can't study the gene's function in an adult. A huge roadblock. The solution they came up with was, well, just brilliant. It's called the conditional knockout. You can think of it as giving scientists a remote control, a genetic on-off switch that they are in complete control of.
And this just changed the game all over again. It means researchers can let a mouse develop totally normally and then flip the switch to turn off a gene in adulthood. Or they can get even more precise and turn off a gene only in the heart or only in the brain. It lets them ask much more specific questions about what a gene does and where it does it.
So this tool has evolved from a powerful but kind of blunt instrument into this highly precise surgical tool, unlocking the deepest secrets of our own biology. It really leaves you with one fascinating question. After completely transforming our understanding of cancer, aging, and so much more, what medical mystery will it help us solve next?